Hello, class. This video is going to review our modern understanding of evolution by natural selection. And this is going to require us sort of going over some of the basic quantitative models used in order to understand how the process of natural selection operates. And so to get started with this, I want to review again what theory of evolution is. And to reiterate, the theory of evolution explains the change in the frequency of inherited characteristics that occurs in populations from one generation to the next. And when we're talking about inherited characteristics, we are talking about genes. And oftentimes, when we're talking about genes, we are talking about alleles, which are variants of genes. And variants of genes change in frequency from one generation to the next, as we have sort of already examined, looking at our population of rabbits. These rabbits carry a certain gene related to coat color, and this gene comes in a couple of different variants. And as the frequency of these variants changes over time, the frequency of the traits is changing over time, and we can observe this population evolving. Now, if we can get at measuring changes in these sort of single locus allele frequencies, so this is just one gene at one particular gene locus, if we can get at measuring changes in those frequencies from one generation to the next, we can start being able to quantify the magnitude of evolutionary change that's happening in a particular trait. And so this is kind of the ultimate goal of getting at our modern understanding of natural selection via a quantitative route. And in order to try to quantify this change or assess this change, we need a model with which to test it again. And in evolutionary biology, this model has classically been summarized as the Hardy-Weinberg equilibrium. And Hardy-Weinberg equilibrium is essentially evolution's null model. Now, if you don't know what a null model is, that's okay. Basically, the model of Hardy-Weinberg equilibrium postulates that allele and genotype frequencies are not changing significantly from one generation to the next. So we might postulate that if a population was in Hardy-Weinberg equilibrium, perhaps this population of rabbits, that the frequencies of these genes would stay pretty constant throughout time, which is, of course, not the example here. Now, this model was proposed way back in the very early 20th century, independently, actually, by two separate individuals, G.H. Hardy, who was a mathematician, and Wilhelm Weinberg, who was a physician. And these two guys came up with the same idea that we can develop a model, kind of based on Mendelian genetics, with which we can test data against to see whether or not populations are evolving or not. And so, again, the assumptions of Hardy-Weinberg equilibrium really require that none of the causes of evolution are occurring. So we have our definition of evolution, and we know that evolution is caused by these four different things. But under the assumptions of Hardy-Weinberg equilibrium, we're basically saying if evolution is not occurring, then there can't be any mutation. If evolution is not occurring, there's no selection. There's no natural selection. There's no sexual selection. There is effectively random mating. We're saying if there's no evolution, probably there's no genetic drift. Or we're saying that population sizes are so large that the effects of drift are pretty much negligible. And we're saying that there is really no migration or really no gene flow. Different populations aren't really sharing alleles. And so an important assumption of Hardy-Weinberg equilibrium, if we're going to use it as a model to test whether or not evolution is occurring, is to assume that all of the factors causing evolution to occur are not operating. And so essentially, major mathematical assessment of this is that if a population is in Hardy-Weinberg equilibrium, the frequencies of genotypes, which are comprised of basically two single locus alleles, they should not deviate from this particular equation that is shown here. And when we're talking about these two single locus alleles, in the context of Hardy-Weinberg equilibrium, they are referred to as P and Q. And what I invite you to recognize, and I apologize for the confusion of this, but this is just the way it's played out. What I invite you to recognize is that P and Q are just like big A and little a, or big G and little g, or big R and little r, or big Y and little y. These are just the two variants of a particular gene that can occur at a particular gene locus. And so what we're basically saying is that if a population is in Hardy-Weinberg equilibrium, the frequencies of these two alleles, and really the frequency of the two genotypes, should not deviate from this particular relationship, which is to say that p squared plus 2pq plus q squared always shall equal 1 if a population is not evolving. These are really the proportions of single locus genotype frequencies expected to occur from one generation to the next when there is no evolution. Now, I will kind of illustrate a basic derivation of how this equation seems to make sense. If we know allele frequencies, so if we know the frequency of P, we can figure out the frequency of Q and vice versa. That's like saying if we know the frequency of big A, we can figure out the frequency of little a and vice versa. If we know these allele frequencies, we can predict non-evolving genotype frequencies under the assumption that there is random mating. And this is very much like saying there is no evolution happening. So therefore, these different alleles are going to pair up at their observed frequencies during fertilization. And so we can kind of look at that example here. And basically the idea is if the frequency of an allele P is equal to 0.4 and the frequency of an allele Q is equal to 0.6, then all matings will involve these two combinations of allele frequencies being involved in fertilization. For example, if this happens in 1,000 matings every generation, P and Q are always going to be pairing up at the same frequency. If that's true, if alleles are pairing up during fertilization at their observed frequencies, then we can generate the probabilities of different genotypes occurring. So, for example, we have in this example here 4 different genotypes. One genotype is, well, there's really 3 different genotypes, but a couple ways to get at one of them. But anyway, we have a genotype composed of 2 copies of P. We have 2 different types of genotypes composed of a copy of P or a copy of Q, and we have a genotype composed of 2 copies of Q. And so if these are our given allele frequencies, let's say P equals 0.4 and Q equals 0.6, then the probability that we will have genotypes that carry 2 copies of P is equal to 0.16. That's the product of 0.4 and 0.4. Likewise, if individuals who carry heterozygous genotypes, or if the heterozygous genotype, if these 2 alleles P and Q are joining up during fertilization, leading to heterozygous genotypes, I should say, then the probability of forming a heterozygous genotype is going to be 0.4 times 0.6, which gives us 0.24. Likewise, 0.6 times 0.4, which is also going to give us 0.24. And so these are the probabilities that we will have these heterozygous genotypes. And then lastly, the probability that we will have 2 copies of Q pairing up during fertilization equals 0.6 times 0.6. That's equal to 0.36. And under the assumptions of Hardy-Weinberg equilibrium, according to our equation, the sum of these probabilities must equal 1. And in this case, they do. 0.16 plus 0.24 plus 0.24 plus 0.36 equals 1. And so this is the basic assumption of Hardy-Weinberg equilibrium. And this might look an awful lot like the binomial expansion you might recall from algebra. P plus Q taken as a squared value is going to be equal to 1. And that is the basis of this equation that we're looking at. If the frequency of P and Q are remaining constant, then these predictions will give us the Hardy-Weinberg equilibrium model as evolution's sort of hypothesis to be tested against. If this model is true, evolution's not happening. If this model is not true, evolution is happening. And as it turns out, the binomial expansion of P plus Q squared equals P squared plus 2PQ plus Q squared, and that equals 1. And thus, this kind of postulates that non-evolving allele and genotype frequencies really should be related. And if they're not, then there's evolution of evolution happening. Now, an important thing to recognize in this is that these different components of the equation directly relate to the frequency of the different genotypes, as we saw in this sort of simple derivation of the equation. P squared represents the frequency of individuals who are homozygous for P. We could think of that as the frequency of individuals homozygous with big A. Likewise, 2PQ represents the frequency of individuals who are heterozygous. We can think of that as being the frequency of individuals who carry copies of big A and little a. And likewise, Q squared is the frequency of individuals who are also homozygous, but for Q, or maybe another way of looking at it as the frequency of individuals who are homozygous for little a. So each of these components of the equation do represent expected genotype frequencies under the assumptions of the Hardy-Weinberg model. Now, I don't want you to get lost in the mathematics of this, such as to say, don't lose sight of the forest for the trees. Applying this type of a principle to actually analyze evolutionary changes is not something that we're going to do in this class, but it's important for you to recognize that this type of thinking is very much the analytical method that evolutionary biologists use today still in order to figure out whether or not populations are evolving, in order to analyze how it is that gene frequencies are changing and whether or not they're changing in a significant way that deviates from this model. Now, this kind of thinking kind of predates something called the modern evolutionary synthesis, but this kind of paves the way for the field of population genetics. And population genetics really provides a quantitative basis for us to understand how it is that evolution by both Mendelian inheritance and Darwinian selection are sort of interacting and leading to the evolutionary outcomes that we can observe in nature. And this concept was most famously first published by R.A. Fisher in this book called The Genetical Theory of Natural Selection, which was published in 1930. And Fisher recognized that there were statistical properties of populations and that these statistical properties were related to these allele frequencies, and that we could actually quantify evolutionary change via inheritance, via Darwinian selection, by understanding, take a quantitative approach to understanding how these allele frequencies and genotype frequencies are changing over time or not. And if they're not changing over time, probably evolution's not happening, and therefore probably this Hardy-Weinberg equilibrium model is true. But if they are changing over time, probably this Hardy-Weinberg equilibrium model is not going to be true. And so this really forms the basis of our modern understanding of evolutionary biology and represents an important point in time when scientists were taking the work of Mendel and Darwin and integrating them together and allowing us to move forward with a really succinct scientific understanding that explains how the process of evolution is actually happening. So we turn the clock back and recall that Darwin and Wallace really first came up with the theory of evolution by natural selection. Now, they didn't necessarily have an understanding of inheritance. They didn't know anything about genes. They certainly didn't know anything about changes in gene frequencies, but they recognized changes in trait frequencies. And of course, we know these traits are directly related to genes for the most part. And of course, we know Darwin theorized that the differences in finch beak morphologies were coming from heritable traits that were influenced, whose changes were being influenced by the environment over time. That is to say, whose frequencies were being influenced to change by the environment over periods of time. And so Darwin was on to this. And so we haven't really talked a lot about what natural selection is in great detail. But basically, what it is a process by which traits associated with increased fitness have a higher chance of being passed from one generation to the next when compared to others. So there's actually a very, very basic definition for what natural selection is. There are a multitude of different traits that vary in the environment. Some of them have a higher chance of being passed on than others, and that is leading to changes in the frequency of these traits occurring in populations from one generation to the next. And as it turns out, natural selection really requires three succinct ingredients. And those ingredients are variation. So we might have a population of beetles. These beetles vary in color, and these beetles vary in size. And then natural selection requires that this variation is heritable. And so this basically means that the differences in color and differences in size in these beetles or within this beetle population can be passed on to the next generation, which kind of is basically assuming that they do have a genetic basis. And then the sort of final caveat is that these different traits have differential reproductive success. And that is to say that certain phenotypes within this beetle population are associated with a higher chance of being passed on to the next generation compared to others. So for example, perhaps we have this hypothetical example here where we have this population of jays, and these jays like to feed on the beetles. And as it turns out, their favorite beetles are the yellow ones for whatever reason. Maybe they're more palatable, maybe they're easier to see, but for whatever reason, the jays will feed on the yellow beetles more often than they will on the red beetles, thus giving the red beetles differential reproductive success. We might start off with a population that has some frequency of red beetles and some frequency of yellow beetles, but over time, because the red beetles have differential reproductive success, the population becomes dominated by red beetles. And so this is really a simplistic explanation for the process by which natural selection unfolds. Variation in populations, that variation is heritable, and different individuals with these different traits have different levels of reproductive success. Now, let's kind of talk about these three ingredients in a little bit more detail. Variation. Well, as it turns out, variation is an emergent property of natural populations, specifically populations that are reproducing sexually. And basically, things like phenotypic traits like height in humans is a classic example, vary quite a bit. We know that not all humans are the same height. And in fact, that height frequency seems to vary with gender. And so this kind of classic image here is showing a whole bunch of students standing out on a football field, and sort of illustrated on this pseudo x-axis are their heights, going from something like 5 feet tall to something like 6.5 feet tall. And this image illustrates, well, how many of these students collectively fall into a whole bunch of different height categories. So we can see that not a whole lot of students are 5 feet tall, and not a whole lot of students are 6.5 feet tall. As a matter of fact, most students on average seem to be close to 5'6" or 5'7", somewhere in the intermediate range. And it turns out when we study variation in natural populations, most often these kinds of traits, these kinds of quantitative traits, seem to be carried at an intermediate level, and individuals with extreme versions of the traits seem to be much more rare. And this is not just true of height, it's true of all kinds of other traits that all kinds of other different species carry as well. Again, we can look at seed mass in these sunflower plants as just another example. Seed mass shown here on the x-axis, going from basically seeds that weigh close to nothing to seeds that are quite heavy. And the frequency of those seeds taken from a population or a sample from a population, most of the seed masses take on an intermediate value, and very, very few are taking on these more extreme values. And so this is kind of the way that we see the distribution of variation playing out across all kinds of traits in all kinds of populations. And as we sort of discussed when we were talking about mutations, phenotypic variation really generally has two causes. There is heritable genetic variation, which is basically caused by the accumulation of different mutations. That's different versions of alleles emerging over time by accident because of mutation. And then there's variation in gene expression that is really influenced by environmental factors. And so there's this really important interaction we sort of talked about between heritable variation that has a strictly genetic basis and variation that we see in phenotypes that is kind of really more based on the way that the environment is causing the expression of these alleles or the expression of these genes to change. And so this is one way we can explain how this variation unfolds. Some of this variation in height might have a genetic basis, but some of it also might have an environmental basis. Different individuals might have been raised in different circumstances with more or less nutrients, let's say, for example, causing them to potentially grow taller. And so the interaction between the genetic basis for their height and the environmental basis for their height leads to the actual physical manifestation of the phenotype that we see in nature. Now, one of the requirements of assessing heritability is that there is a correlation between the phenotypic trait values of parents and their offspring. And basically, we could use size as a pretty good example of this. Let's say we look at this graph on the left, that the x-axis represents the average size of parents, or really the average size of parents, and the y-axis represents the average size of their offspring. And if this particular trait related to size is in fact heritable, there should be a pretty tight relationship along these these two axes. And this is kind of illustrated over here. And basically, if a trait is heritable, then we tend to see a pretty tight one-to-one relationship between the mean trait value of parents and the mean trait value of offspring. And we tend to signify heritability with this variable called h squared. And in this particular situation, h squared equals 1. And if h squared equals 1, we're basically saying the trait has a 100% genetic basis. It is 100% heritable. And this might not look too confusing to you, but or this might look familiar to you, but this value of heritability really represents the slope of the line. And so in this case, the slope of the line is equal to 1. And therefore, there's a tight one-to-one relationship between the mean trait value of the parents and the mean trait value of the offspring. So if, for example, we took a particular parent whose trait value is somewhere here around this big star, probably if heritability is equal to 1 or close to 1, the mean trait value of the offspring would be pretty close to that value as well. And so we can almost make a prediction about what the size of the offspring is based upon what the size of the parents is. That's if heritability is equal to 1. Contrasting scenario would be when heritability is equal to 0. And in this case, the line simply goes flat across parallel to the x-axis. And in this case, we see a sort of scatter of points on this plot, indicating that this trait is under really has no genetic basis directly at all. Everything about the trait is really being influenced by the environment. And so this is kind of how we assess heritability and get an understanding whether or not a trait has a genetic basis, and then kind of how much of a genetic basis does it have. And obviously there are a multitude of values between 0 and 1 that heritability could take on. And so as heritability starts to decrease from 1, we start to say, okay, there's more and more and more and more environmental influence affecting these traits and less of a genetic base. Heritable traits undergo this thing we call differential reproductive success under the process of natural selection. And so we can imagine a situation where we have the mean size of the parents, again, and the mean size of the offspring, and they are correlated with one another in a very nice, tightly knit way. And this sort of suggests if from one generation to the next, this keeps unfolding in this way, and there's really no change in the frequency of the parent trait value and the frequency of the offspring trait values, we're kind of saying there's no evolution happening. However, if there is natural selection going on, then we might see the value, this mean trait value of the parents change. And likewise, the mean trait value of the offspring change. And so, for example, if this particular proportion of the population does not survive, but this particular proportion does, we're going to see that mean trait value shift quite a bit. And we're going to see that the mean trait value in the offspring went from being whatever it was here to becoming whatever it is here. This population has evolved. It has undergone differential reproductive success. Individuals with the larger trait value, the larger size in this example, had a higher chance of reproducing compared to those the smaller size. And those traits were being passed on to the next generation because they're heritable, but they're being passed on at disproportionate frequencies because of differential reproductive success. And so in essence, phenotypic traits associated with a higher probability of inheritance are the ones that are going to be associated with something we call increased fitness, because they have a higher chance of being passed from a parental generation to an offspring generation, or a second generation, if you. So we talked a little bit about this thing called fitness. Effectively, fitness is really the currency of natural selection. It's the basis upon which natural selection is operating. And we can talk about fitness in a couple ways. The simplest way to talk about it is in the context of something called absolute fitness, which is signified by the letter big W. And absolute fitness, it could be quantified in different ways, or there could be different proxies, if you will, for how we quantify absolute fitness. But most simply, it's quantified as an individual's probability of survival to maturity times the expected number of offspring that they might have. And so this is saying, what are their chances of actually making it to a reproductive age where they're possibly going to have offspring? And then how many offspring might we expect them to have based on some sort of a priori knowledge? And this gives us an idea about the absolute fitness that individuals carry. And absolute fitness really depends upon these things we call fitness components. And these fitness components really require that there's reproductive and developmental success during any stage of a species life cycle. And so this image on the right sort of shows a typical or a hypothetical species life cycle. We could start from, let's say, the stage of gametes. And so if gametes are able to be successfully involved in fertilization, that contributes to fitness and leads to the formation of zygotes. And if those zygotes are viable, that contributes to fitness. And if those zygotes develop into adults, that contributes to fitness. And if the separate females and males of the population have successful matings, that contributes to fitness. And if they are able to become parents because their offspring are healthy and viable and successfully, if they're able to become parents and they themselves become reproductively mature, that gives them something called fecundity, which allows them to start producing gametes again, and the whole cycle repeats itself over and over. And this is happening amongst not just our species, but in this example, all sexually reproducing species are undergoing this kind of a life cycle. And during these various stages of the life cycle, they are able to continue gaining fitness. And this is something that allows us to quantify differential reproductive success. So one of the most common ways that differential reproductive success can be quantified is by looking at something called relative fitness. And this can be evaluated by comparing the reproductive successes of different genotypes. So imagine this hypothetical example on this plot here where we have three different genotypes, big A, big A, big A, little a, and little a, little a. And on the x-axis is their frequency, or is the number of generations that are passing, going from 0 to 300 generations, and the frequency of these different genotypes that are occurring over this period of time. And so initially speaking, all of these genotypes have a frequency of 500 individuals, but we can see as the generations progress, these genotypes are not reproducing at an equal rate. And as it turns out, it looks like big A is having the highest reproductive success, big A, slightly less reproductive success, and little a, significantly less reproductive success. And we can measure this thing called relative fitness, which is instead represented by the little w letter. We can measure the relative fitness of these genotypes by simply dividing the frequencies of each one at the final point in time by the one that has the highest frequency. So for example, big A has the highest frequency after 300 generations. It went from having 500 individuals to having 447. So the population size hasn't declined that much. And so we can take 447 and divide it by 447, and the relative fitness of that genotype is equal to 1. Likewise, we can take the frequency of the genotype big A, little a after 300 generations, which is 204, and divide that by the frequency of the big A genotype, which has the highest frequency, and that gives us a relative fitness of 0.46 for the big A, little a genotype. And similarly, we can take the frequency of the little a, that's a mistake here. Let me fix that for you. The little a genotype, which is 35, and divide that by the frequency of the big A genotype, which has the highest fitness. And that gives us a relative fitness of 0.08 for the little a genotype. So by comparing the relative fitnesses of these different genotypes, we can sort of come up with a basic way of quantifying and comparing their fitnesses. And this is important for understanding how it is that natural selection might be favoring one genotype over the next. And in this particular case, it appears to be that the big A genotype is being favored much more than the others. And in this context, there's even information to suggest that there's something beneficial related to carrying a copy of big A. In fact, carrying 2 copies of big A seems to be the most beneficial scenario. Carrying 1 copy of big A is slightly less beneficial, and carrying no copies of big A is even less beneficial completely. And over time, if we projected this out to say 400, 500, 600 generations, we might even expect that the little a genotype would disappear from the population. And this, in fact, is something that we think about happening under a process called selective sweeping. And this has to do with something we call selection coefficients. There's some few different ways that we can calculate selection coefficients using relative fitness, but basically a selection coefficient, which is represented by the letter little s, the selection coefficient is a measure of the strength of selection. And so in this particular situation we just looked at, we might say that there's a high selection coefficient favoring big A, and a lower and lower selection coefficient favoring these other genotypes. One way we can look at it. Basically, we're saying the strength of selection is favoring this big A genotype. And so we can also look at how the strength of selection, as quantified via a selection coefficient, is really influencing the rate at which a beneficial mutation becomes fixed in a population. And by fixed, we mean that this mutation goes from a very low frequency to a very high frequency. And the selection coefficient kind of is a way of looking at how many generations does it take for this mutation to spread through the population. And this is something we call, again, a selective sweep. And as we can see, we have 3 different scenarios here with 3 different selection coefficients. A selection coefficient of 0.01 is comparatively weak, a selection coefficient of 0.02 is quite a bit stronger, and a selection coefficient of 0.05 is stronger yet again. And in fact, this is just about as strong as the force of selection is known to be in nature. If we actually look at real selection coefficients, they're usually not this high. But what I would like you to see here is that as this selection coefficient is increasing, the rate at which this mutation is reaching fixation is going up. When the selection coefficient is equal to 0.01, even over the course of 500 generations, the mutation has not really come close to reaching fixation. When that selection coefficient is higher, that is to say the strength of selection for this allele is greater, then the frequency of this mutant allele is reaching fixation in something on the order of about 400 generations. When the strength of selection is over twice as high, we can see that the frequency of this mutant allele is reaching fixation in something closer to about 200 generations. And so the point here is that the strength of selection matters, and it can very much dictate the rate at which certain alleles become more and more common within populations. Now, an important caveat of this is to recognize that, isn't this like sort of driving genetic diversity out of populations? And the answer is yes. Really, really beneficial alleles under strong selection will tend to become completely dominant within populations. And so at a particular gene locus, there might be only one variant of an allele that is present because that allele apparently is so beneficial. But that could be a bad thing because there could be certain situations where all of a sudden the environment changes and only having one copy of an allele present at both the 2 different homologous chromosomes, for example, is not favorable. But anyway, this is a simple model. It's a hypothetical example. And the realistic situation is, or the reality of it is, over the course of this many generations, 500 generations, it's very likely that other evolutionary forces are going to come into play, either new mutations or the influences of things like genetic drift. So this question about, well, how might this lost genetic diversity be restored? Probably it could be restored by the introduction of new mutations into this population. But this is basically a simple model to show that we have observed in nature very beneficial alleles becoming very, very frequent, very, very quickly, depending on the strength of selection. Now, the number of generations that it takes for an allele to reach fixation under this so-called positive selection model is very much proportional to the strength of selection. We have 2 contrasting versions of this story here. One such story is that we have a selection coefficient of 0.05, and we have, again, this mutant allele. And we can see that under this type of strength of selection, that it takes only about 88 generations for the frequency of this mutant allele to increase from about 10% in the population to about 90% in the population. And so that's a relatively fast period of time if selection is sufficiently strong. When selection's weaker, as shown on the example on the right, let's say the selection coefficient equals 0.02, it might take something closer to 220 generations for the frequency of this mutant allele to increase from 10% to 90%. So this is not a whole lot different than what we see with our selective sweeps. The strength of selection matters, and it directly influences the amount of time it takes for a beneficial mutation to spread within populations. There are some statistical ways we can look at this. Basically, this value of 4 divided by the selection coefficient is representative of the number of generations approximately that it would take an allele frequency to increase by 80% in this way. And we know that the probability of fixation, or it's been estimated that the probability of fixation is about 2 times the selection coefficient. But this is not necessarily always completely realistic because other evolutionary forces can step in and change this projection. Again, 500 generations is a very long period of time. The other evolutionary forces that we know exist are likely to emerge at some point or another within the course of 500 generations, especially if an organism's generation time is long. So as we sort of have seen from these examples, that selection coefficients really can be used to predict the rate at which allele frequencies are going to evolve. And this points towards something very important, which is that the rate of evolution is very much proportional to the strength of selection and the amount of genetic variation. Now, again, don't get lost in the math, but basically, we're again thinking about this allele being represented by P, kind of like we saw in the Hardy-Weinberg equilibrium. And then we're thinking about this other version of the allele that's 1 minus P, which is really just Q. And basically, the product of P and Q represents the amount of genetic variation that a population carries. And of course, we can think of S as representing the strength of selection. And so the product of the strength of selection and the amount of genetic variation are really directly correlated to the expected change in the frequency of a given allele, which is known as delta P. And so larger amounts of genetic variation, higher selection coefficients lead to higher expected changes in allele frequencies, and vice versa. And we can kind of get this idea that we can make a prediction about what the frequency of an allele might be based on its expected change and its frequency at a particular point in time. But the really important take home of this mathematical modeling is that the rate of evolution really depends upon how strong selection is and how much variability there is in populations. If there's not a lot of genetic variation within populations, there's probably not going to be a very fast rate of evolution, or not much evolution at all. Again, if the strength of selection is weak, there's not going to be a very fast rate of evolution. Imagine if S was equal to 0, well, the expected change in the frequency of P would be 0. Imagine if, we'll just leave it there. And so basically, the evolution of allele frequencies follows a handful of different contrasting trends. We've really been talking so far about something called positive selection, and this is selection that really favors one beneficial allele. And so just imagine that we had some mutant allele we call A2, and we have 3 different scenarios where the frequency of this allele starts off at whatever values, be it 0.1 or 0.4 or 0.7. In all of these scenarios, if we modeled them in a computer simulation, over some period of time, depending on the strength of selection, these alleles would reach fixation. And the graph on the left is kind of showing that genotypes that carry this beneficial allele have increasingly higher fitness, or rather the genotypes that carry this beneficial allele have higher fitness. This is in contrast to a different model, which is known as overdominance. And overdominance is actually selection that favors relatively equal frequencies of 2 different alleles, which we call heterozygotes. And so this is a scenario where if we start off with a very low frequency of some mutant allele, or a very high frequency of a mutant allele, or a sort of intermediate frequency of this mutant allele, in all these different simulations over time, a relatively intermediate frequency of these alleles is going to be favored. And this is again kind of illustrated in this graph here, where the heterozygotes seem to have the highest fitness compared to the homozygotes. And this is something that turns out to be important in a whole lot of different environmental contexts, because again, individuals who carry two different alleles at a particular locus are often capable of doing two different things compared to individuals who carry two copies of the same allele at a particular locus. And this turns out to be true for folks who are heterozygous for the beta hemoglobin protein, which I'll talk about in a second. There's something that's far less common called underdominance, but basically this is selection that favors the homozygous genotypes. And so again, in our model here, we could start off with these mutant allele frequencies, 0.3 or 0.4 or 0.8. And under this type of selection, the homozygous state is going to be favored, and much in contrast to overdominance, here we see that the heterozygotes have the lowest fitness. And this is kind of weird, but it could be because different individuals have chromosomal incompatibilities, or different individuals have experienced local adaptation. So these homozygotes might not do very well at mating with these homozygotes. These homozygotes might be locally adapted to certain circumstances that these homozygotes are not, and vice versa. And if these homozygotes try to mate with these homozygotes, it might cause something called outbreeding depression, where their heterozygous offspring have lower fitness than the homozygous parents. This also kind of points toward the idea of adaptive landscapes. Now, this is very much theoretical, very much sort of just being used as an analogy, so to speak. But basically, this idea kind of projects how allele frequencies are changing under different kinds of selection. And so in these models, allele frequencies are kind of have been said to shift from low fitness valleys towards high adaptive peaks. And these three different graphs here are the same, well, very similar to the three graphs we see here. And these graphs on the bottom are kind of showing these adaptive landscapes. And so basically, we're looking at three different genotypes here on each of these x-axes, and we're looking at their relative fitnesses here on these y-axes, much as we saw with these different graphs. And we are here now looking at the frequency of this particular mutant allele, A2, which is the same mutant allele that we were looking at on the y-axis, or the y-axes of these different graphs. And what we're seeing is represented by these arrows is the strength of selection. So in the case of positive selection, there is a selection pressure that is driving fitness to increase as the frequency of this mutant allele is also increasing. In contrast, under overdominance, we're seeing selection pressures kind of coming from two opposite sides, wherein we see that selection is favoring an intermediate frequency of this mutant allele, thereby favoring the frequency of these heterozygous genotypes. And so just to reiterate, this down here is sort of like the low fitness valley, and this here is sort of like the high adaptive peak. In contrast, in this situation, we see this here as being a low fitness valley, and this here being a relatively low fitness valley, and this here being the sort of high fitness peak. Highest fitness is achieved when the frequency of the mutant allele is close to 50%. And in contrast, underdominance is kind of like where the frequency of that allele being close to 30 or 40% is the low fitness valley, and the high adaptive peaks are achieved when that allele frequency is either 0 or 1, depending on the type of genotypes that are carrying that allele frequency. I should say, depending on the alleles that a particular genotype carries. Of these three models, positive selection obviously is thought to be pretty important because beneficial mutations can spread through populations pretty rapidly. But let's talk again a little bit about overdominance. And overdominance is really important because it's something that maintains genetic diversity within populations. And we know that genetic diversity within populations enables them to have a better chance at dealing with processes of environmental change with environmental variability. And this sort of process of natural selection favoring overdominance is called balancing selection. just has some other names like balanced polymorphisms. But basically, this is an idea where there's a balance between the two alleles that occur at a particular locus. And this is what drives overdominance. And so we, again, could have this model where we have a particularly beneficial allele or an allele. It's a mutation. And it starts off at a either really low frequency or an intermediate frequency or a high frequency. But over time, it turns out that an intermediate frequency of this allele is much more beneficial to the population than a dominant frequency or a high frequency of this allele. Because while carrying a copy of A2 is beneficial, it's also beneficial to carry a copy of A1 at this particular locus. And this leads us to the concept of heterozygosity. Selection is favoring the frequency of heterozygote individuals in this population. And heterozygosity really leads us to a measure of genetic diversity in a different way under the assumptions of so-called Hardy-Weinberg equilibrium, where we could say 2 times the frequency of P times the frequency of Q is how much genetic variation that a population carries. Saying P is like saying A1, for example. Saying Q is maybe like saying A2. They're the same thing. And so the idea is that under overdominance or under balancing selection, individuals who carry different alleles at a single locus have higher selective advantages than individuals who are homozygous at those loci. And probably the most classic example of this is seen in this particular gene leading to the formation of beta hemoglobin at this particular locus. And under this scenario, we see that sometimes actually selection is maintaining deleterious alleles because of this heterozygote advantage, because of balancing selection. So we know that there's this gene that codes for beta hemoglobin, and it's represented by 2 different alleles. One's called big A and the other's called big S. Again, I know it's kind of inconvenient that we're constantly shuffling around the different letters that we use to represent alleles, but that's what scientists do. But what we classically know is that humans who carry 2 copies of the S allele, who have SS genotypes for the beta hemoglobin gene, are probably going to generate a very, very terrible and harmful disease called sickle cell anemia. Now, if you don't know what this is, basically, sickle cell anemia is a situation where individuals' red blood cells take on these really bizarre shapes. And these bizarre shapes of these blood cells tend to clog blood flow. They tend to clog veins and arteries. So this is kind of like what a normal red blood cell looks like here, normal hemoglobin. And this is what a sickle blood cell looks like. It's very much reduced in size, and it does not pass through veins and arteries nearly as easily as a normal red blood cell. But individuals who are homozygous at this locus for this gene happen to have these sickle cell blood cells. And if they're not treated properly, this condition can actually be lethal. As it turns out, carrying only 1 copy of the S allele is beneficial in a very interesting way, and that is because carrying 1 copy of S confers resistance to a very, very lethal disease called malaria, which is caused by this plasmodium, which is transferred by mosquitoes. And so we can look at these 3 different genotypes in populations that are carrying these different alleles, and we can see that there are differences in their survival. There are differences in their fitness. And as it turns out, the heterozygotes have the highest fitness. Those individuals who carry 1 copy of the normal allele, which is big A, and 1 copy of the mutant allele, big S, have the highest fitness. Individuals who carry 2 copies of big S obviously are going to have really low fitness because they're probably going to have sickle cell anemia. But why is it that we see individuals who carry 2 copies of big A having lower fitness than individuals who carry a copy of big A and a copy of big S at the same time? It's because carrying 1 copy of big S confers resistance to malaria, whereas individuals who don't carry any copies of S are susceptible to malaria, and so their fitness is a little bit lower. And what we kind of interestingly see if we compare these 2 maps of where malaria is most frequent in the world or most common in the world in Central Africa is that the frequency of the S allele is at its highest in areas where incidences of malaria are highest. And that's kind of interesting. And probably there's selection favoring the evolution or favoring the persistence of this big S allele in these environments where malaria is really common. So in this situation, we're looking at a very real example of why balancing selection is important. Why is it that there is this sort of balanced polymorphism or this overdominance? Carrying two different copies of an allele at a particular locus can be more beneficial than only carrying one copy. In some cases, carrying one copy can be downright lethal, and in other cases, carrying one copy could just be less beneficial than carrying two different copies. And so this kind of points toward this idea that even though there are deleterious alleles that exist, sometimes they still are maintained via natural selection because of different environmental pressures. And this leads us to another interesting ideas about mutations, which is that they actually can be expected to occur at predictable rates, despite negative selection or despite selection against them. And this leads to something we call mutation selection balance. Now, please do not confuse balancing selection with mutation selection balance. These are two different things. The idea behind mutation selection balance is best described via something we call the bathtub analogy. And the bathtub analogy basically postulates this scenario where if we can imagine a bathtub and all of the water in the bathtub represents a gene pool, that is the sum of different genes that exist in a particular population. And in this example, let's just say we're only talking about genes at one locus. And so we have this collection of different genes that can occupy this locus. And we can think of the drain of this bathtub being left open so that the water is draining out. And this drain is sort of analogous to the force of natural selection, removing these harmful mutations from the gene pool, taking them out. These mutations are not they're not conferring any benefit to the individuals that carry them. So selection is driving them out of the population, much as a drain would take water out of a bathtub. But at the same time that we have the drain open, we also have the faucet running. And this faucet running is analogous to new mutations being reintroduced back into the population, which happens. And so as it turns out, despite the fact that selection is very good at removing harmful mutations from populations, those harmful mutations seem to continually reoccur again and again and again at predictable rates. Now, often these rates are very, very small, but they exist nonetheless. We have a sort of mathematical representation of this, basically where if there is a predictable mutation rate, which we know many mutations do occur at predictable rates, and there is a certain strength of selection against this mutation, the quotient of that mutation rate to the strength of selection against it is proportional to the minimum frequency at which that mutation will occur in the population. And that's called p-hat. But anyway, the point to understand, again, don't get lost in the math. The point is that certain mutations will continue to persist within populations, albeit at very, very, very low frequencies, because even though selection is good at removing them, those same mutations might seem to reappear again and again and again. So in all of these examples we've been talking about, in terms of models of natural selection, we've generally been looking at single locus traits and single locus trait frequencies in particular, and sort of handful of different ways that we can predict their change or their changes from one generation to the next. But we also know that natural selection is influenced by selection pressures acting upon what we call quantitative traits. And these quantitative traits are continuous. And these quantitative traits are not single locus traits. These traits are what we call traits that have undergone polygenic inheritance. That is a fancy way to say that there are probably multiple genes involved in forming these traits. For example, height is almost certainly a quantitative trait. And these traits, instead of analyzing their frequencies at a single locus, we instead analyze their frequencies across a continuous scale, a continuous quantitative scale. And so this is very much like, again, how is the frequency of different heights distributed within populations? How might the frequency of different seed masses be distributed within certain populations? In these examples we're going to look at, we might say, well, how is the frequency of different pigmentation in these different hypothetical fish populations being influenced by natural selection? And so we might have three different phenotypes within this fish population, a sort of very light colored phenotype, or a very dark colored phenotype, or maybe a sort of intermediate phenotype. And we have these three kind of contrasting scenarios where selection's either favoring one extreme type of trait, perhaps maybe the light colored fish, or maybe natural selection's favoring the two extremes. So really dark colored fish are favored and really light colored fish are favored, but the intermediate forms are not favored. Or in contrast, yet again, situations where very much the intermediate trait is being favored. And these types of models of natural selection have very specific definitions. And so the first example where we look at fitness for the light colored fish, hypothetically speaking, being favored is something we call directional selection. And basically, we can look at this on this graph here where as the value of this trait increases, that is, as the trait is becoming increasingly lighter and lighter and lighter in color, the survivability of individuals who carry the trait is going up and up and up. And so the highest chances of survival are being associated with one extreme. And we can look at the cartoon here, and that really nicely illustrates how maybe in the first generation, there's sort of this normal distribution of these different fish phenotypes, and the light colored fish aren't actually very abundant at all, but somehow the environment is favoring them. And in the next generation or in subsequent generations, the frequency of light colored fish is going up. And the frequency of these other fish colors are going down. And likewise, again, some subsequent number of generations later, we see that the frequency of the light colored fish has come to dominate the population. And this is a case where the extreme phenotype is increasing in frequency, sort of illustrated with these two plots here. If we wanted to analyze this more statistically, we can see that this is the original distribution of the traits, and the average value is shown by this little red caret. But over time, that frequency is slowly starting to shift to the right. That average trait value is starting to shift toward an increasing value, as suggested by this plot. So that's something we call directional selection. Again, this is in contrast to another model where the extreme phenotypes are being favored and the intermediate phenotype is not. And in this case, we see that we again have trait values here, and really, really low trait values are favored and really, really high trait values are favored, but the intermediate trait values are not. The intermediate trait values are associated with the lowest survival compared to the other two. And if we look at our fish example again, we can start off with the same sort of distribution as we saw in directional selection. And over time, selection is now favoring increasingly either darker colored fish or increasingly either lighter colored fish in subsequent generations. And then some number of generations later, we see that both dark colored and light colored fish have come to dominate this population in terms of their abundance. And this is kind of represented again with these graphs where over time, opposite phenotypes are increasing in frequency. We went from having a sort of normal distribution of these things to having their distributions flattened out. And ultimately, we might end up with a bimodal distribution in this scenario. This thing we call disruptive selection. It's also known as diversifying selection. And sometimes this could be an initial step in the process of speciation. Over time, if there's reproductive isolation between dark colored fish and light colored fish, they might tend to have higher chances of associating with one another, and they could diverge into separate lineage. And then this other model that comes up is something called stabilizing selection. And stabilizing selection is a scenario where selection is favoring intermediate trait values along this scale. And so we might, again, have our trait values here on the x-axis. And it turns out that individuals with the most intermediate values of this trait have the highest chance of survival. And so once again, if we start with our sort of normal distribution of our fishes, our fish phenotypes over time, fishes with the sort of intermediate trait are being favored by selection, so that in subsequent generations, there are higher frequencies of fishes with the intermediate color, let's call it gray, and lower frequencies of the dark colored fish and lower frequencies of the lighter colored fish. In fact, we see in this example, the lighter colored fish have left the population altogether. They've become locally extinct. And then over many, many more generations, we see that the relative frequency of the intermediate fish color is significantly higher than that of either the darker, or the lighter colored fishes. And in our graphical examples, we can see how this intermediate phenotype from one generation or one set of generations to the next, I should say one generation to the next, the frequency of this intermediate trait has really been favored, such that we see our distribution on this plot going from being sort of shaped like a bell curve to becoming much more narrowly shaped like this. And so this is an example of stabilizing selection. And this example is something that is very much played out, we think of as dictating the size of the heads of human babies that have the highest chance of successfully undergoing childbirth. And so that's kind of the classic example of where small heads are not favored as much because they don't facilitate large brain size compared to larger head sizes, but larger head sizes have a harder chance of undergoing birth successfully. And so we kind of think that an intermediate head size has been favored in humans up until this point in time because of the process of stabilizing selection. So that's it for this lecture. I know it was a lot of information, but try to digest it, and we'll catch up next time.